Okay, we are in Chapter 8 on genetics. In this first set of slides, we'll be covering the structure of DNA and RNA, the concept of DNA replication in a prokaryotic cell, though this does translate very nicely to what's happening in a eukaryotic cell. Then we'll look at transcription and translation. There are key concepts for these that will carry over throughout the rest of the semester. This would be understanding DNA strand orientation. So what is the structure of a DNA nucleotide and how it orients itself with this concept of 5' to 3'. So we'll look at that in a little bit more detail. Understanding transcriptional language, so being able to use a codon chart and knowing what that a codon is. And essentially the whole process from DNA to proteins, so understanding transcription and translation and being able to do both what I call the shorthand version, which is just taking a sequence of DNA and converting it through transcription and translation into a protein polypeptide product, but then also the concepts behind this as well, and the key enzymes involved in the steps. Before we jump into genetics, there are some terms that should be discussed first. Hopefully you guys are familiar with some of these things already, but if you're not, then let's review a few of these. All right, so what is a genome? The genome would be a collection of genetic information inside an organism or inside a cell. So let's say the collection of genetic info, let's say in an organism. Usually we're talking about DNA here, deoxyribonucleic acid, which holds the pivotal information to develop all the proteins and regulatory machinery in the cell. But we can also be talking about, actually I'll say, DNA in the form of chromosomes, which holds the essential genetic information. But there are other forms too in a prokaryotic cell. This can be also in the form of plasmids. These are small circular collections of genes that are non-essential. Plasmid. But we could also be talking about the RNA. This would be all of the stuff we'll be talking about here, messenger RNA, transfer RNA, ribosomal RNA, but also a collection of regulatory microRNAs and small interfering RNAs and other things that all could be part of this genome. But all RNA stems initially from DNA, so really DNA is the foundational concept of this term genome. What is a gene? A gene is the encoding region . The encoding region on DNA. I know we've talked about this before. Let's just draw our little figure here. So in a prokaryotic cell, remember they have single chromosomes, right? This is double-stranded. This is a double-stranded DNA molecule, and then on this huge genome, or on this chromosome, sorry, there are specific regions that encode for proteins. And for prokaryotic cells, I've already talked to you already that these guys collect multiple genes together and stick them in one region. We call these things operons, a collection of genes. Already my thing is slowing down. Collection. I know it's painful, guys. Of genes is called an operon. And so each of these genes or gene segments is a region of DNA that will then be transcribed into mRNA, and further translated into a polypeptide, which is then folded into a protein. All right, so this is the process of transcription, this is the process of translation, and this is just folding or post-translational modification. So we have genes on DNA that are the coding regions. All the rest of the stuff here, kind of between these operons or between these gene segments, used to be called once upon a time junk DNA, but that is definitely not true anymore. You'll still hear that term every once in a while, but we now know this stuff in between is really important in regulation of cell processes and also of transcription and translation. So what would the gene product be? In technical terms, I'm going to say a polypeptide. Poly means many. Peptide is in reference to amino acids. But in reality, what we are talking about here are proteins. All right, so a gene product is a protein. And remember that enzymes are also proteins. So when we're discussing proteins, we're also, and most importantly, talking about the production of enzymes. What is genetics? This term is the study of the function of DNA, right? Study of DNA. What it does, how it works, replication, transcription, etc., right? The study of DNA. And so what is the study of DNA? Well, there are two primary components to studying DNA. That's this part here. All living cells must do two things to survive. These are what we call the basic tenets of genetics. The basic tenets of genetics. One, DNA must be replicated in order for that cell to reproduce, right? So duplication of the original DNA molecule, and this occurs only once. So replication, this occurs once in a cell cycle, right before binary fission, otherwise known as cell replication, right? So the DNA must be replicated in order for that cell to replicate. So we'll start with this process here after we look at DNA structure. But then also at all times during that cell's existence, that DNA is transcribed into RNA and then translated into that functional protein product downstream. So this is where we will end these first sets of slides is on transcription translation. But let's start with looking at the structure of DNA altogether. So what does this thing look like? DNA is deoxyribonucleic acid. It has these three basic ingredients. So there is a phosphate group, phosphate, connected to a 5-carbon. This is the 5th carbon here, 4. And if you notice in biochemistry, they like to name or number their carbons, and we number them like this with a little prime number. So here are the 5 carbons in this ribose sugar. Carbon number 1, 2, 3, 4, and 5 connected to a phosphate group here. There's an oxygen essentially at the top of this ribose sugar. And then a nitrogenous base. This nitrogenous base is called a base. This nitrogenous base is connected to that 1' carbon, and the phosphate group is connected to the 5' carbon. So here's this sugar phosphate core with a nitrogenous base attached to it. And you'll note that this nitrogenous base is what gives DNA its sequence. So when you're talking about the DNA sequence, you're not talking about the sugar and the phosphate. Specifically, you're discussing which nitrogenous base is bound at each of these 1' carbons for these nucleotides. Now, one of these guys here, from here up and over, this is called a nucleotide. One nucleotide contains a nitrogenous base, a ribose sugar. I keep saying ribose. This is deoxyribose, right? So there is no hydroxy group here on this 2' carbon. So this is technically called a deoxyribose sugar, and this phosphate group. So these 3 components are what makes up a nucleotide, and a nucleotide is also called, where am I going here? Multiple nucleotides connected together would be a single strand of DNA, right? So this is a polymer of nucleotides. That's the term I'm looking for, polymer. And each nucleotide is connected here between the existing 3' carbon, connects the next nucleotide at this phosphate group. This bond here is called a phosphodiester bond. You don't need to know that, but this bond is what's formed. So this is what's formed between adjacent nucleotides. It's this bond here between the existing 3' carbon here and the new nucleotide's phosphate group. Again, here's another nucleotide, phosphate, 5', 4', 3', 2', 1'. Here's that 3' carbon, and here is the connection from this nucleotide to the next nucleotide, right? So all these dotted lines are indicating a bond that's formed from one nucleotide to the next. And I really want to emphasize this understanding so you really see this connection and the requirement for this available 3' carbon on this nucleotide here to bond this next incoming nucleotide at this phosphate group. Now, when these nucleotides exist all alone, so when they're kind of floating out in the cytoplasm, you'll find this actually connects with 3 phosphates. So it's a sugar with a triphosphate and a nitrogenous base on it. This might sound familiar because we've talked about that exact same structure for our energy molecule ATP, which is also a nucleotide. So in its free form, this nucleotide has 3 phosphates on it. When that nucleotide bonds to another nucleotide in a strand of DNA, those 2 phosphates are essentially released, and this 3rd phosphate is bound to that 2' or 3' carbon. So this is called a sugar phosphate backbone. If you note, the sugar phosphate is exactly the same all the way up this strand. And if you were to look at this this cartoon image of a double-stranded DNA molecule, what you see in the red and the blue, that would be considered this sugar phosphate backbone. That's what this drawing shows right here. So this is sugar phosphate. It's exactly the same all the way around. And then these rungs that you see on the inside of this ladder, the rungs are these nitrogenous bases on the inside. And again, this is what essentially gives the flavor for DNA. This is what determines the sequence, is the nitrogenous base on the inside. So let's look for a second though. I just was showing you one strand of DNA. This is bound to another strand of DNA to form the entire double-stranded DNA molecule, right? Double-stranded means there's 2 strands that are bound together. Now we call this type of relationship, if you notice that this sugar phosphate backbone here is exactly the same as over here, except it's flipped mirror image and it's inverted, right? This is called anti-parallel Anti-parallel arrangement or orientation. This term orientation is an important one. It's essentially the orientation, how these things are arranged in relation to one another. And this is where your numbering of the carbons becomes very relevant. So if you notice in this strand that we just looked at with this sugar phosphate, the 5' carbon is essentially like on the top of our little house we have here. Here's 5, 4, 3. 5, 4, 3. 5, 4, 3, right? 5, 4, 3. So you would indicate that by saying this strand is in the 5 to 3'. There's 3 down here. 5 to 3' orientation, or I could even write it like this. 5 to 3. Right? That indicates the 5' carbon is essentially at the top of your nucleotide and the 3' carbon is on the bottom. Now, in a double-stranded DNA molecule, this strand here would be called the complementary strand. Complementary, or the complement. This complementary strand is in the anti-parallel orientation, which means the 3' carbon is at the top. 3, 4, 5, right? Let's write our numbers in here. Again, here is one nucleotide, but it's anti-parallel, so it's flipped in mirror image and upside down, right? 3, 4, 5. So this we would indicate from 3 to 5', as you see here, or you could say the complementary strand is anti-parallel. In this case, it's 3 to 5, right? So if one strand is 5 to 3, the other strand will be 3 to 5. You would draw it 5, 4, 3 like this, and this one will be drawn upside down, 3, 4, 5 like this, okay? Both of these, again, have the same components. So here's your phosphate that's bound to this deoxyribose sugar. We already wrote these carbon numbers, 5, 4, 3. Here's carbon number 2. Whoops, 2. And here's carbon number 1. And just as we saw before, that 1' carbon is connected to the nitrogenous base, right? So for DNA, let's see if I can get my pen over here. Nope, I don't know why it does that. So for DNA, I wonder if I could do it over here. Nope. All right, over here. For DNA, there are 4 nitrogenous bases that DNA can have. That would be adenine, thymine, cytosine, and guanine. This is where you get your A, T, C, and G. And there is a rule of binding, right? So when complementary strands come together, they form these bonds at the nitrogenous bases. These are called base pairs, a pair of bases or nitrogenous bases. So the rule of pairing A's or adenines will always pair with or bind with 2 hydrogen bonds to thymines, and the same goes backwards. Thymines will always bond with 2 hydrogen bonds to adenines. Cytosines will always pair with or bond with 3 hydrogen bonds. So this is a stronger bond here with guanines, and same vice versa. Guanines will always bond with 3 hydrogen bonds to cytosines, and you can see that stuff here. A's and T's, C's and G's, C's and G's, A's and T's, right? It doesn't matter if you have a T here or an A here, the rule of binding will always be the same. And these are called complementary nucleotides, right? An adenine is complementary to a thymine. A guanine is complementary to a cytosine. This is another picture to show you these same things. And if you were curious about this rule of binding, well, it's based on the biochemical structure of these 2 different nitrogenous bases. And you can see that the guanines and cytosines match up perfectly with their nitrogens, right? And the ability to form these hydrogen bonds at each of these major groups. There's someone walking down the street with some toilet paper. I'm afraid he's going to get attacked. Okay, here's the C's and G's and T's and A's. And then again, here's our sugar phosphate backbone. D stands for deoxyribose sugar. Don't forget our numbering system here. It's a 5-carbon sugar. So there's 1, 2, 3. Whoops. Well, I don't know what they did here. Yeah, so 5 carbons with an oxygen group in there too, to have there's your 3', 4', and 5'. Okay. To have your phosphate groups here, so sugar phosphate all the way down. This would be, if the 5' carbon is way up here on the top, that means that this is the 5 to 3' orientation this way, and this would be anti-parallel complementary, so therefore we are 3 to 5' orientation, kind of going in the other direction, right? And then again, here's our adenines and our thymines with our 2 hydrogen bonds formed down there. So you should be able to work through this one. We'll do it together as a discussion during lecture time. If the nucleotide sequence of one strand of DNA is this, what is the sequence of the complementary strand? And then the second part here, I definitely want you guys to be able to do this. Draw the following double-stranded DNA molecule, labeling carbon positions and indicating orientation. So practice that one on your own. For our lecture, we'll do this one in class also. And for our online lectures, we'll do this one on our Zoom conferencing, and I'll have you guys submit this one too. Do the process of DNA replication. So note on this video, I'm just going to work through these slides first. If I were doing this in person, I would be both going back and forth with the slides and the whiteboard. So for this purpose, I'll do the whiteboard portion after I go through these slides. So you'll have a visual, me working it out by hand on here too. So let's talk through this process. What do these 2 terms mean? DNA replication for prokaryotic cells is bidirectional and semi-conservative. Semi-conservative is a term that's applied to DNA replication across all species. This concept means that during the process of replication, one old strand is passed on to both of the daughters in this process. And this one I can kind of draw out for you. You'll see many examples of this too. So if this is the old strand, I'm just going to draw both in red. Remember, we just looked at DNA strand orientation. So we know if we looked at this up close in person, we know that one of these things is like in the 5 to 3' orientation, and the other strand is in the 3 to 5' orientation because of the rule of complementary binding or complementary pairing. So don't forget that. These strands are complementary to each other. They're not identical. During the process of DNA replication, where these guys are this whole double-stranded DNA molecule is going to be copied and made into a whole new copy. So we have 2 that are made, or 2 new DNA molecules, double-stranded DNA molecules that are produced at the end of replication. Semi-conservative means one of these is going to serve as a template for one of the daughter cells. So here is that, for example, that 3 to 5' strand. It will serve as a template. That means just like you have a template when you're making a copy of something on a copy machine, right? That would be your original. And then the stuff that comes off of that copy are the copies, or in this case, going to be the new strand. So let me try to change this color just so I can show you the difference here. So if that's the old strand, then this is the new strand that's made as a complement to that template, right? And this would be the new strand in the 5 to 3' orientation. So one old and one new, that should say 3. And over here on this side, we have the other strand acts as the template. Here is the 5 to 3 as the template, or the old, right? The old strand, or we'll also call it the template. Hopefully you understand this term template. And the new strand is this guy on the inside this time in the complementary orientation 3 to 5. And that would be the new strand, right? New strand. So this is what semi-conservative means. Semi is partial. So the newly generated replicated double-stranded DNA molecules have conserved one old strand for each of them, and for each of them a new strand has also been added, or bound complementary, right? So there we go. This term bidirectional is unique to prokaryotic, actually that's not true. Yes, bidirectional is unique to prokaryotic cells, and it's this concept we have a circle that we're working with instead of eukaryotic cells, which is linear. Here we have an opening that occurs at one spot. This is the origin of replication. And from here, replication will ensue in both directions. So you'll have replication events occurring in this direction, but also in this direction. So I think I have some pictures of this. You can see that in this next slide here. Again, here's that origin of replication for this double-stranded DNA molecule. These two molecules will open up, and they will each, as I showed you in that last picture, each of those double-stranded DNA molecules will become templates for new strands to be made on top of that, and that replication occurs in both directions. So you'll have it as that circle opens, essentially replication will begin this way, but also replication will begin this way. So you see that in this picture here, there is we call this a replication fork on both sides of this opening circle. Sort of hard to visualize here, but there are some videos that will help you show you this process so that that concept of bidirectional replication makes sense to you. And here's again a picture of the semi-conservative concept where you have the old strand in the blue in this picture and the new strand in the red being generated from that template. In this one we see the old strand in the red and then the new strand in the blue dots being made from this template. Here's a picture. This is actually an electron micrograph where you can see both of these little points right here are called replication forks. It's where that circle essentially has been open. In this case, it looks like it's open almost halfway, and there's replication happening in both directions. So from here, sort of all the way down this way, and it's continuing down, and will keep going all the way till it gets to the very end of this circle. And once you have that happen, again, here's another picture of exactly the same thing, where essentially this was the original, that circle would be the original DNA molecule. This has already opened up. Replication has already occurred down in this direction and in this direction. So you can see the old and the new here, the old and the new here. And this replication will continue all the way down until the bottom of the circle, essentially till both of these replication events meet each other down here at the bottom. It'll look like this, where there will be a nick and a release of those daughter molecules. This is one more picture to show you the same concept. Again, here's that circle and the origin of replication, one point on this double-stranded DNA molecule. Here's the initiation of replication, but again, it's going to occur in both directions, so over to the right and over to the left. And you can see that here with this initiation of this complex. This is the replication machinery that a cell will use. And there's two complexes that are formed, one that will go in one direction and one that will go in the other direction. right? And so you'll see that this replication event occurs in both directions, and it ends up looking something like this. I just want you to keep this in the side of your mind, this concept that replication occurs in both directions of the circle. But for the rest of this conversation about replication, we're really just going to focus in on one of these directions, one of these replication events that are occurring over here. And we'll look at, and even this becomes a little bit complicated. So we'll look at what's happening right at that site of replication as this whole machinery essentially opens up this double-stranded DNA molecule and replicates both of those strands, the template strand and the template strand here, one in the 3 to 5 prime orientation and one in the 5 to 3 prime orientation. So this process, and again, I'll draw all of this stuff out for you, but I'll draw some of this stuff as we kind of go through, just so you can, just so I can follow this thing along. So that process begins at the origin of replication, as mentioned already. So here's your double-stranded DNA molecule, and there is one site, and we'll call it the ORI site. This is the origin of replication. And it's a sequence of DNA, a sequence of those nucleotides that is where the binding of these initiation enzymes can occur, right? That's why there's one site here. And this is found only one spot on a chromosome and also on plasmids. All plasmids must also have their own ORI site, or origin of replication, in order for them to continue to be replicated. So from here, we have an opening of the 2 strands, the double-stranded DNA molecule. And there are a couple of key enzymes involved in this pathway, the topoisomerase and helicase, and I don't really care which one you put first or second, but we'll just keep it like this. Essentially, remember that this double-stranded DNA molecule, actually, maybe you don't remember, this is wound in a helical orientation, and also usually this is coiled up pretty tightly in order to conserve space essentially inside that cell and to prevent and to help regulate the function of the DNA molecule. So in order for replication to occur, in order for these strands to open, essentially the unwinding needs to occur. Or if you can imagine, like if you separate a rope by its strands and you just start at the top, and you know a rope is very tightly wound too, right? It's helical. If you separate a rope by its threads and start pulling it open, essentially what you're going to end up with is a more tightly coiled rope at the end. So the topoisomerase is essentially, it releases the tension of that coiling DNA. And so we're going to just say it unwinds DNA, but really it's about reducing the tension, or releasing the tension as that DNA strand is being opened up. And you didn't see me at all. I was just using lots of hand motions to describe that. So let's just kind of write this out here. So here at the origin of replication, let's go down the bottom, and we'll write it like this. So here is an ORI site, and here's your double-stranded DNA molecule still wound up. So we're going to focus on, let's say that this is the site of origin. So it's that double-stranded DNA molecule has opened up. There's all these nitrogenous bases that have been exposed, right? These are all indicating those nucleotides, specifically those nitrogenous bases. And we know that each of these strands is going to act as a template. But we also remember that they are in different orientations, right? That one is in the 5 to 3 prime orientation and one is in the 3 to 5. Let's just say that this one is in the 3 to 5 prime orientation. And so that means that its complement, right, the new strand is going to be 5 to 3. So down here, this is the template. So our template is 5 prime over here and 3 prime over here. So its complementary new strand is going to be in the opposite orientation. Okay, so over here on the side, let's change some of our colors. I know I'll have to change this every time we go into a new screen, so we'll put over here on the side, our unwinding occurs here. Number 1, topo, not a good color. Topo, I, sa, mer, ace, unwinds. Right, so here's our topoisomerase. It's unwinding that DNA. Change that color. That one's painful to look at. This is probably going to be bad too. Here we'll put a helicase. Helicase is the second enzyme here. A helicase unzips that DNA. In other words, it breaks open those hydrogen bonds that were once keeping these nitrogenous bases together. So number 2 would be helicase. C-A-S-E. And we just term this unzipping, but it breaks hydrogen bonds. Unzips. Okay, so topoisomerase unwinds, helicase unzips. Now we have these open double-stranded DNA molecules. These guys don't like to be open. They don't like to be exposed, so they will immediately close back up again right away. So in order to prevent that, there are these key molecules called single-stranded binding proteins that will come along right here. Single-stranded binding proteins. Oh, this is a bad color too. Will come along and essentially bind to, oh my gosh, bind to the strands to keep them open. So number 3, single-stranded binding proteins equals prevents re-annealing. Oh man, this is so slow. Okay, prevents re-annealing. That means prevents these strands from binding back up again. And then our key initiation event would be the binding of an RNA primer with this key enzyme called RNA primase. Some people call it a DNA primase, but we're going to keep it with this terminology, which is how I learned it. So the binding of an RNA primer using an RNA primase enzyme. Now we're just going to focus on one of these two strands because DNA replication always proceeds in one direction. And this is a very important note. DNA replication always precedes replication. I'm sorry, I'm so slow. Always proceeds in one direction, which is always, at least for the new strand, the newly synthesized strand is always synthesized from the 5' to the 3' orientation. And the reason is, you'll see it in just a minute. This 3' carbon that we saw in that early slides, that 3' carbon is always needed in order for a new nucleotide to come and bind, right? So replication always proceeds in the 5' to 3' orientation, and this is the orientation of the new strand. This is an important rule to remember. Of the new strand. Parentheses. Okay, So replication always proceeds in the 5' to the 3' direction of the new strand. So let's go back down here to our little drawing. Remember that both of these guys are going to act as templates. Both of them will need to be replicated. But this rule is always true. So let's look at how this will ensue or proceed. For this strand, what is the orientation of the new complementary strand? Well, the complementary strand is going to be, let's turn that to red. It's easier to see. The new or complementary strand of this guy, new strand, will be in the 5 to 3 orientation in this direction, right? Complementary. This is fine. So this is the direction of the replication fork. This machinery is opening up this double-stranded DNA molecule in this direction. So with this top strand, you can have continuous replication as that double-stranded DNA molecule continues to open. In fact, we call this the leading strand, the leading strand. So the leading, L-E-A-D-I, you can see my pen marks, I'm actually writing this, leading strand is where the newly synthesized strand is in the 5 to 3' orientation in the direction of the replication fork. The leading strand is where the new strand is in the orientation of 5 to 3' in the direction of the replication fork, right? So this allows continuous synthesis of the newly synthesized strand. However, let's look at this other template down here. This also needs to be replicated. But the problem is that this is in the opposite orientation, or anti-parallel orientation, if you recall. So this one, the template is in the 5 to 3' orientation, which means the new strand is going to be in the opposite, right? 5 is on this side, and 3 is on this side. And remember, we said that replication always proceeds on the new strand from 5 to 3. That is no different down here. But because the orientation, in order for this new strand to be synthesized from 5 to 3, then you might have already guessed that synthesis occurs in the opposite direction. The problem here is that this is the direction of replication. These guys are continuously opening up these new molecules, right? And although we know that on this side over here, there is another replication fork, which you can recall, but what's happening to these nucleotides here that are continuously being opened? If replication is occurring to the left on the lagging strand down here, we call this the lagging strand, the lagging strand is the strand whose orientation essentially requires replication to occur in the opposite direction of the replication fork, okay? So we're going to see how this whole step-by-step process of replication, it occurs exactly the same on both of these strands. However, because of the direction of replication, on the leading strand, I'm just going to follow through this process one time for the leading strand, and then we'll talk about how on the lagging strand, this process from this point on will occur many, 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 many times as these double-stranded DNA molecule continues to open in this direction. So if that doesn't make sense now, just wait. We'll see a bunch of pictures of how this whole process works as we go through. But for now, let's just focus on the leading strand and this step-by-step process. So the same is true for both leading and lagging for 1, 2, and 3. First, topoisomerase unwinds the DNA. 2, helicase unzips the DNA. 3, single-stranded binding proteins bind to hold the strands open and prevent reannealing. So step number 4 is the binding of an RNA primer at the initiation of replication. So the RNA primase lays down an RNA primer. Let's try to do this in green. And we're just going to focus on the leading strand. Again, I'll show you how this occurs on the lagging strand in just a little bit, but let's do this next enzyme here. All right, so here is our RNA primase that lays down an RNA. Oh my gosh, I've got to figure out my technology. A very short fragment of RNA. And this will prime, this is your number 4. Again, I'll write all this stuff out on the whiteboard without such horrible, painstakingly slow computer delays. This is number 4, the primer. P-R-I-M-E-R. All right, our primer is laid down, and this is RNA primer, not DNA, this is RNA. And this is essentially just to prime the new strand so that a 3' carbon is now available for the binding of new nucleotides. So it's a very short 3 to 5 base pair, or a 3 to 5 nucleotide primer that is bound complementary to the start of a replication event, and it initiates replication by providing that 3' carbon for the next complex. Okay, let's go forward from here. Everything's going to get erased. Now we have the whole elongation process occurring, which is the binding of the DNA polymerase number 3. And this is the key enzyme, and it's an enzyme complex, but we're just going to focus on this one enzyme itself. DNA polymerase 3 binds and adds new nucleotides to the existing 3' carbon of that primer, and then it continues this elongation event essentially until the end of a replication cycle. So let's look at this picture here. This is the DNA polymerase here. It's a very large complex, and it sits upon that template strand at the site of replication. So this DNA polymerase 3 proceeds, and it does only one thing. It brings in nucleotides and adds new nucleotides to the existing 3' carbon. So if you recall our DNA strand orientation and the carbon numbers, here is carbon number 1, carbon number 2, carbon number 3, number 4, and number 5 for a nucleotide here. And here is that deoxyribose sugar here with that 3' carbon, and a phosphodiester bond is going to be formed right here, where the removal of a hydroxy group with those 2 phosphates, those guys go away essentially and form a new product, and then we have a phosphate bond formed between this 3' carbon and this phosphate group with this incoming nucleotide. So this is the key function of this enzyme, is to form this phosphodiester bond between a new nucleotide and the existing 3' carbon of an existing nucleotide. Now, the nucleotides that are brought in are complementary to those on the template strand, right? So this polymerase sits upon this entire thing and allows complementary nucleotides to come in and bond, and that forms these hydrogen bonds here. But it specifically will form this phosphodiester bond at that 3' carbon. So this is the relevance of that primer that we just talked about. The primer is responsible for initiating this process by laying down the first few nucleotides to provide that 3' carbon so that the polymerase can come and just continue this elongation process. This polymerase is really key. Also, just so you know, it has proofreading machinery because it's so big. Sometimes an accidental incorporation of the wrong nucleotide. So for example, maybe here instead of a C, a T was put in. This actually causes a bulge in the DNA because the binding is not, or the pairing is not exact. So this polymerase can actually stop and rewind essentially and remove any mistakes that were made and continue that process. So it has some proofreading capacity. On the leading strand, As long as the direction of synthesis is towards the replication fork, this process can continue indefinitely, and well, this process can continue until the end of that entire replication cycle, right? And again, note that the template strand here is in the complementary orientation as the newly synthesized strand here. At the very end of this process, so once that whole half of the circle is replicated, then the very last couple steps occur. Recall that we had started with a primer that was not DNA. This is an RNA primer. And so at the very end of a replication process, a new enzyme called DNA polymerase number 1 will come in and essentially take off that RNA primer and replace it with DNA. But now we have a tiny gap between this new DNA and the newly synthesized strand, and so the ligase is an enzyme that comes in and seals that last little gap to form a complete circle. The ligase will become important as we talk about this whole process on the lagging strand too. So that was the step-by-step process for the leading strand. Again, I'm going to go onto the whiteboard in just a second to show you this again, and I'll explain on the whiteboard how the lagging strand compensates for the reverse direction of this strand orientation. And when I come back, we'll pick up from here to explain this image here. All right, guys, this is the process of DNA replication. So we've already heard about this process on the slides, and we'll just draw out this whole thing here. So I'm going to draw it so that the replication fork is over on this side, but I do challenge you to be able to draw this with your replication fork anywhere. So you should be able to draw this with your replication fork up here or down here or over on this side. It shouldn't really matter. So we'll also draw it so that our leading strand is up here and our lagging strand is on the bottom. Again, you can redraw this any way. If you recall, the leading strand, the template, for the leading strand template, remember that replication always proceeds in the 5 to 3' orientation for the new strand. It's always built essentially from 5 to 3. So in order for us to understand which is which and essentially to figure out the strand orientation, for the template or the old strand here, it's going to be complementary to this one. So we know that the template strand is from 3 to 5 in this direction. So if you were to look at each of these nucleotides here, each of these nucleotides would have their 5' carbon on this side and their 3' carbon on this side. If you need to understand this in a little bit more detail, go watch some videos on this so it can really show you, highlight these strand orientation concepts. But this also means for the lagging strand down here that the template is complementary, of course, to our other strand. So this would be 5' is on this side, 3' is on this side. So the 5' is over here and 3' is over here for these nucleotides on this strand. So I'll show you first what's happening, again, step by step on the leading strand, and then we'll discuss what's happening on that lagging strand at the bottom. So let's start with, I'll try to color code this as best as I can if I can remember what we just did with the slides. So for DNA replication, recall that this begins at the site of origin, the origin of replication. We have 2 key enzymes involved in essentially opening up this double-stranded DNA molecule. I'll write them over here. So number 1 is our topoisomerase. And we're going to say this one unwinds. The topoisomerase unwinds. I know that's a little bit light, but that's okay. And recall that we mentioned that unwinding essentially means it releases the tension on that coiled DNA. So as you're opening up this double strand, that double-stranded DNA just keeps getting tighter and tighter. So the topoisomerase comes and relieves that tension of the coiling or the coiled strands. Number 2, helicase. helicase. And this breaks those hydrogen bonds. So we call this unzipping, right? So it breaks those hydrogen bonds between those nitrogenous bases. And then we have the opening of the double-stranded DNA molecule, and recall that we need to make sure that we prevent the reannealing of these strands. So we have the binding of our single-stranded binding proteins on the backs of this sugar phosphate backbone. So number 3, oh, my marker. Number 3 is the single-stranded binding proteins. Just a second. I have more markers. Trying to get some out. Single. Oh, it's my board, not my markers. That's the problem. Single-stranded binding protein. Oh my gosh, can't see any of this stuff. Single-stranded oh, look at that. Single. Prevents reannealing. And for this term reannealing, to anneal means to pair back up with their hydrogen bonds. So to prevent reannealing means to prevent the closure of this double-stranded DNA molecule back up again. This is an important concept, as we'll learn in the next process, transcription. Reannealing is vital for the health of a DNA strand. So in this process here, we are trying to, or the molecules here their intent is to prevent that so that we actually develop 2 new strands altogether, or 2 new double-stranded DNA molecules altogether at the end. Okay, so we have our steps 1, 2, and 3. Now we begin the actual process of DNA replication. For the leading strand, we call this leading because this new replicated new strand is going to be built in the direction of the replication fork. So here's our steps. This is step number 4. This is the RNA primase, which is an enzyme. Lays down short RNA primer. Okay, so there's a very short segment that's built here, and so that 3' carbon is available. Remember that this is going to be built complementary or anti-parallel to the template strand. So if this is 3 to 5, this is the 5' end of these nucleotides and the 3' end to these nucleotides, or yeah. So this is an available 3' carbon. The purpose of this primer is to prime the new strand, right? This is the new strand. And for the leading strand, although the template is 3 to 5, the new strand for the leading template over here, the new strand will be from 5 to 3 in the direction of the replication fork. That is the most important thing to understand, that we are always talking about in the direction of the replication fork. So there's the RNA primase. It lays down a short RNA primer, and this allows for that 3' carbon available now for replication. Now, while I'm at it, let's just draw it on this lagging strand too. Now remember that we have the opposite orientation. So we'll draw this lagging strand again in the opposite direction. The same step applies here, the development of this RNA primer on the lagging strand, but the orientation will be opposite. So let's draw it over here. Again, this is step number 4, RNA primase. Lays down that RNA primer, so here is that short 3 to 5 base pair RNA primer that's laid down. Again, for the availability of that 3' carbon here, 5' is over here, and this is the direction of replication. That should say 5. Okay, so this is the direction of replication here. Because of this rule, DNA replication always occurs from the 5 to 3', or at least the new strand is built from 5 to 3'. So here's 5 and 3. The reason it's going backwards, again, just to remind you, is based on the orientation of this template on the bottom. So we saw a picture of that DNA polymerase and how it lays down on top of the entire complex and just allows the continuous addition of new nucleotides only by adding on to this existing 3' carbon. So let's see, step number 5 would be the elongation event. So here we have now the ability for DNA polymerase. I'll just draw it over here. Really got to work on these markers, don't I? All right, so here is our DNA polymerase. Now I'm going to elongate and essentially just continue to elongate from this 3' carbon. So it sits on top of this complex and moves down this way, adding on nucleotides in this direction, right? Just continues to add them on in this direction, continuously adding on to that available 3' carbon. And this will move on and continue indefinitely, essentially, on the leading strand until it reaches the end or the halfway point of that circular chromosome. This is step number 5, DNA polymerase 3. This elongates the new strand always in the same orientation from the 5 to 3'. So by adding you can kind of see that DNA polymerase 3 elongates the new strand by adding nucleotides to the 3' carbon. All right, so always requires that 3' carbon and continues to elongate by adding new nucleotides to that 3' carbon. And this is how you get those complementary nucleotides bound, right? So if this is an A, you'll have a T coming in. If this is a C, you'll have a G coming in as your new nucleotide. Now, again, for the leading strand, this will occur continuously until the end of the entire replication cycle. For the lagging strand, it's going to occur also. So we have here that 3' carbon. So we'll have an elongation event where the same thing, step number 5, this DNA polymerase will come and bind and add those nucleotides in this direction, right? So we have that 3' carbon continuing the elongation event in this direction. Now we're going to actually come into a problem because eventually you'll run out of nucleotides. And recall that you have these nucleotides continuously opening in this direction. So if this is the start of a replication event, you have a whole bunch of new nucleotides that have just been open and exposed in that direction. So for the lagging strand, this process will occur essentially over and over and over again. So let's say that this was the origin of replication, right? We had these nucleotides exposed. Here at the leading strand, that's fine. We have the initiation of replication here, and it just continues in that direction. As new nucleotides are opened, they are essentially replicated. On the lagging strand, let's say that this was the origin. We had initiation occur here, and it has to go in the opposite direction, but all of these nucleotides are being opened up here. So what happens on the lagging template is that as more nucleotides are exposed, this process, the RNA primase lays down an RNA primer, and DNA polymerase III elongates, will actually occur over and over and over again as more nucleotides are exposed. So let's just draw in here an example of earlier on when these initial nucleotides were exposed. Then we probably had another primer here, right, with that 3' carbon. And we had another polymerase coming and elongating in this direction here, in that direction. And so we have these short fragments of replication, and we have a term for this because there is continuous over and over and over again. These short segments are being created in this direction. These short fragments are called Okazaki fragments. So these short bursts of DNA replication occur on the lagging strand, again, because replication occurs in the opposite direction of that replication fork, that these short segments are created here on the lagging strand over and over and over as that strand continues to elongate. These are termed Okazaki fragments. So each of these segments here would be an Okazaki fragment. Now, again, at the end of a replication process, so let's go back up here at the top. At the end of this whole replication event, after elongation is completed, we still have this RNA primer here, and then multiple times here on the lagging strand, that needs to be essentially taken out and replaced with DNA. And so here we come. Herein enters our next, let's see if pink works. Kind of works. All right, so here I'll draw on the lagging strand because it's easier to draw. So here, after this whole replication event occurs, we have, can you see that? Not really. Just trying to get a color that you can see. I'll draw it in blue. Okay, so here we have at the very end of this process an enzyme called, this is step number 6, DNA polymerase number 1. This goes and removes and replaces. The RNA with DNA. Okay, so RNA polymerase 1 will come in to each of these different RNA primers that are here, kick them off essentially, and as it goes through, it will fill in the gaps with DNA nucleotides. So now at least we have all DNA. And then the very last step, there are going to be these gaps here and here in the DNA that's generated from this portion here and between all of these different fragments. And so the last step will be something called DNA ligase. So this would be step number 7. This will be the ligase. Essentially, we call it sealing the gap. This is like the glue. Seals the gap in DNA and completes the entire replication event. So these are all the key enzymes involved in this process. Let's just walk through it one more time. Okay, walk through it one more time. So again, at the origin of replication, we have the initiation of both the leading and lagging strand replications, right? For the leading strand, the template is 3' to 5' orientation because the new strand will be 5' to 3' in the direction of the replication fork. So this is called a replication fork here, where we can draw all these enzymes, and you guys should be able to practice this one on your own. So here again, number 1, topoisomerase essentially unwinds that coiled up DNA strand. It's not exactly true, but releases the tension on the wound strands. Step number 2, helicase comes and unzips or breaks those hydrogen bonds between the nitrogenous bases on complementary strands. Step 3, the binding of the single-stranded binding proteins to prevent the re-annealing or the closing back up again of these strands. Now we have open leading and lagging strands. Let's just look down here at the lagging strand. Step 4, the initiation of a replication event with the laying down of an RNA primer using the RNA primase enzyme. This occurs in always the development of the new strand from the 5' to the 3' orientation. For the lagging strand, that means this is going, shoot. I'm not sure why I did that. Okay, from the lagging strand, that means that this replication event is occurring in the opposite direction of the replication fork. For the leading strand, that means that this replication event is occurring in the direction of the replication fork. Step 5 is the binding of the DNA polymerase 3, right? So this complex comes and lays down at that primer and begins to elongate by adding nucleotides to that existing 3' carbon. So here again, that replication event occurs on the lagging strand in the opposite direction of the replication fork. But regardless of which strand you're looking at, always just keep in mind the concept of strand orientation. So if you were to just look at the event itself, this process here is exactly the same as this process up here. Exactly the same, because the orientation is the same if you were looking at it like this. So essentially the replication events are going like this, right? The opposite direction. But the process is the same, where this polymerase continues to elongate by adding new nucleotides to that 3' carbon, binding complementary nucleotides to the template. And now we have our 3' to 5' template and our 5' to 3' complementary template. So this again, just to reiterate that concept of semi-conservative replication, there is your old strand and your new strand. Here's your old strand and your new strand, right? So semi-conservative. This replication process will conserve the old strand for both of the new double-stranded DNA molecules that are being generated. Step 5, the elongation will continue all the way until the end of that replication cycle on the leading strand. For the lagging strand, this process will continue until it hits the next excuse me, Okazaki fragment. There's another one. Excuse me. Once this DNA polymerase butts up against the next Okazaki fragment, essentially that complex will fall off. Now we have to go back in and remove all of that initial RNA on both the leading strand and the lagging strand. This occurs really just once here on the leading strand, where this RNA primer is removed and new nucleotides are put in with the use of this enzyme here, number 6, which is DNA polymerase 1. It replaces it replaces the RNA with DNA. On the lagging strand, this will occur every place that there is an RNA primer. Finally, we have the last enzyme involved, the DNA ligase, which seals the gap, and that will occur up here, and it will occur between all of these Okazaki fragments on the lagging strand. Summarize again the process of DNA replication. We see in this image, this picture from your textbook, the process of, sorry, there's some people outside. The process of DNA replication. This is showing you with a replication fork essentially up here at the top, where the direction is going up. But as I said, I want you guys to practice drawing this where the DNA replication fork is on the left or on the bottom or on the right, with the leading strand and lagging strand inverted. So practice this a few different times over. And in fact, I'll have that as a posted assignment as one of your practice sessions here, is to draw this in at least one other direction so that I can see that you guys are really thinking about the concept of orientation. All right, so here is step number 1, that topoisomerase essentially unwinds the coiled up tension here with the DNA. And the helicase here, step number 2, would break those bonds, the hydrogen bonds here, or it unzips, right? So topoisomerase, step number 1, DNA helicase, step number 2. Here are the single-stranded binding proteins that bind to both the leading and the lagging strand, just to keep the strands from re-annealing. Next is the DNA polymerase 3, right? And this is showing you the leading strand here, the lagging strand here. Oh wait, whoops. Where's the primer? Okay, I don't know why it has it as step number 5. That's definitely backwards. Okay, so your textbook needs a little bit of help here. But In order for DNA polymerase 3 to exist, it needs to have an initiation event here, which is the RNA primase laying down RNA primer. So please reorder that in your book. Here, let's just say again, step number 4 would be, as we've written it, RNA primase lays down an RNA primer. You don't see it here on the lagging strand because this is just showing you that way down over here was the origin of replication, which is where that RNA primase would have been active, laying down the RNA primer, which is what it states here. But on the lagging strand, you can see it here. The RNA primase lays down that very short RNA primer. Just a few nucleotides is all that's needed to provide that existing 3' carbon. So that's step number 5. The DNA polymerase 3 can come in, bind to that 3' carbon, and begin elongating by adding new nucleotides to that 3' carbon. So here we see a difference between the leading strand and the lagging strand. For the leading strand, this elongation event can be continuous in the direction of that opening replication fork, versus on the lagging strand, this is developed in short segments because as that, I keep wanting to show you guys with my fingers, but as this replication fork continues to open in this direction, the replication events occur essentially over and over and over again on this lagging strand in that backwards direction. And so these short fragments that are generated here are called Okazaki fragments on this lagging strand, and that replication event will occur in these short intervals. At the end of the elongation event here, and also here at the very end of this process, then we have the removal of that initial RNA primer with a DNA polymerase 1, which is not listed here. And then finally, those gaps between Okazaki fragments on the lagging strand, and the final gap that's on the leading strand at the very end of this process is filled here with the DNA ligase, right? And so that would be in our description, step number 7. This is another picture from a previous textbook version. So this is again showing a different replication fork, in this case going in the left side, as you can see up here, this replication fork. And you can see again, the top one is the leading strand, and this is showing you somewhere downstream, so you don't see that initiation event here, so you don't see that RNA primer laid down on the leading strand, but you do see it on the lagging strand. Here is the start of that replication, that RNA primer, and the lagging strand, of course, is going in the opposite direction. So here's the RNA primer that's being laid down by the RNA primase. Step number, what are we on? So here's topoisomerase and helicase. You don't see topoisomerase in this one either, but that's okay. Helicase, you don't see the single-stranded binding proteins, but they do exist here. So this would be step 4. The RNA primase lays down RNA primer. Step 5 is the elongation event that's occurring here and here for both. This is the DNA polymerase 3 that adds nucleotides onto the 3' end of that existing nucleotide. And remember that it's in the opposite direction of the replication fork for the lagging strand and in the direction of the replication fork for the leading strand. At the end of this short segment on the lagging strand, we have DNA polymerase 1 that comes in and removes the RNA primer and essentially replaces them with DNA nucleotides. And then that final step, the DNA ligase, will come and seal that gap. And then just one more visual for you. This one does have all the key ingredients in order. Again, topoisomerase, helicase coming and unzipping single-stranded binding proteins. On the lagging strand up here at the top this time, we have our RNA primer being laid down by the RNA primase. In this case, it's called the DNA primase, but it's the same thing. Lays down that initial primer. DNA polymerase 3, or polymerase alpha in this case, comes and elongates and continuously adds those nucleotides in the direction of the replication fork for the leading strand and opposite direction for the lagging strand. That final step is the replacement of those primers with DNA using DNA polymerase 1, and then ligase comes and seals that gap. So some terms you should become familiar with. That replication fork is essentially that where that DNA is opening with the topoisomerase and helicase to provide available nucleotides. We've discussed leading and lagging strand. You should be able to draw all these on your own now. And then this term Okazaki fragments relating to those short segments of replicated DNA on the lagging strand. So a few questions. How does the leading strand differ from the lagging strand? You should use strand orientation to answer this question, and we'll go over this during our lecture period. This one here, the second question, I do want you guys to work on your own, and I'll have this as the place you can upload this on Canvas. Draw a replication fork showing the events of replication, include strand orientation and the direction of replication. So all this stuff is important for you guys to really get down, and again, really think and focus on the concept of strand orientation. So as best as you can, draw this in a direction that you haven't seen yet, or at least in a direction that we didn't physically draw out. So draw it with a replication fork on the left or on the top or on the bottom, and draw it with your strand orientations different than what we have done already. But you should be able to draw this step by step all the way through. So that's it for replication. I'm going to pause the video here and pick back up again to talk about transcription and translation.